It's really a pleasure to see you, and I see many known names and faces, but I also see a lot of new ones, which really makes me happy. It's exciting to have you all here, and maybe more will join as we get started. About this exciting, really, PTC Pivot HD study that's just rolling out now in Europe and has already started in the United States. So many of us, or some of us, were at least in Bologna just a couple of weeks ago and heard a little bit about this study from the stage. But hopefully today we can manage to get a little bit more in maybe even more lay-friendly language, Sarah.
So let me see if I can share the screen. Excellent.
Okay, my friend. So then please present yourself, Sarah, because many may know you, but maybe not all.
So thank you for inviting me to present. I'm going to present on behalf of PTC about the Pivot HD study. I'm a professor of neurology based in London at the UCL Queen Square Institute of Neurology. I have worked in Huntington's for about 25 years, doing a huge amount of research and doing a lot of work to try and find potential disease-modifying therapies for this disease. I still see patients every week and share with you all the urgency that we need to be able to develop and test potential disease-modifying therapies in the context that, as Dina so really beautifully and very movingly presented at EHDN in our clinical trial session about the kind of whack-a-mole feeling of the different trials that have been paused, stopped, halted. So I'm going to tell you about a new study sponsored by PTC. So I don't need to tell this audience about Huntington's disease. We know Huntington's disease is rare. It has orphan disease designation. And as all of you know, the symptoms typically begin in middle age. And I don't need to also tell this audience about the symptoms of Huntington's disease in terms of motor change, personality change, behavior, and cognitive changes. And for that reason, we want to test and develop therapies that really target all of these areas by targeting as much of the brain as possible. And one of our biggest challenges in therapeutics is delivery. And because an organ the size of the human brain, in the studies in mouse models and non-human primates and pig models, none of these models of Huntington's are the human disease, and none of them have a brain that really is even comparable to the human brain. So many approaches are targeting Huntington. And why is that important? Well, numerous animal studies, including genetic studies, have shown that if you lower the level of the mutant Huntington protein or total Huntington in adulthood in animals, you change the course of the disease. Now, there are over 50 publications with different approaches. There are 3 really beautiful genetic studies where you use genetic methods to switch down the level of the Huntington protein in mice. There are many approaches with something called small interfering RNA and antisense oligonucleotides. And now we come to the potential of small molecules. And what we mean by the word small molecules is something you take orally. And the reason the field really is focusing on Huntington lowering, because this is a genetic disorder. You inherit the Huntington gene, and we know that that toxic protein that the gene makes causes the disease. We know that if you lower the level of that in animals, you reverse the disease. And it's lowering the level of the harmful protein by interrupting, in this case, the message. And this is the approach that I'm going to talk to you today. And also, importantly, it's a tablet. So what makes the PTC518 molecule unique? It has had more than 10 years in development, and it works at low doses. It has few to no off-target effects. And what we mean by that is affecting other genes or proteins or messages in the brain that you didn't want to be affected. And in the animal studies and in cells, it looks like this is very specific for Huntington. And the studies so far in healthy volunteers have shown that Huntington lowering with this therapy is safe in the timeline of the healthy volunteer study. Importantly, it's given by mouth every morning. So it's a new Huntington lowering drug in clinical trials. And so why do I think this is important? I think it's important because it's oral. It's an oral tablet. And it's an oral tablet trying to do things that other approaches are also doing that are targeting the brain directly. But the potential for families and patients is that an oral tablet will be much easier for them. It has the potential to treat everyone in the world, including very poor countries where the access to be able to have invasive treatments is less. So that's why I think testing an oral therapy like this is important. And this has been designed for Huntington's. So it's taken by mouth every day. And as I said, we hope that it may treat Huntington's everywhere in the body. And the trial is going to test whether it lowers Huntington in the human brain. And the drug is called an investigational drug. And what that means is its safety and whether it lowers Huntington in the human brain has to be tested in a clinical trial. So how does this oral tablet work? So it works in cells through a mechanism that's quite complicated, where it's affecting splicing. But what I will explain it simply to you is the tablet interferes with the Huntington message. And what it does with the Huntington message is it puts it causes something to be inserted into the Huntington message, which basically tells the Huntington message to stop making the Huntington protein. It's basically a stop sign. And that is added into the Huntington message, and that results in the Huntington protein being not made because you have a reduction in the Huntington message. So basically, it's affecting the Huntington message that makes the protein. By putting this stop sign into the Huntington message, you get less of the protein. That's the way this works. So the PTC518 has been tested in HD mouse model, and it was tested to see if it lowered Huntington. And this data shows here that what's, I think, important is the lowering in blood is similar to the lowering in brain. And the reason that's important in an oral tablet is because you want an oral tablet that gets enough into the brain, which is the place that we want it to work primarily, without having very high levels in your blood, because this is total Huntington lowering, and we don't want very, very high levels of total Huntington lowering in the periphery because of wild-type Huntington. So the important thing here is that it is lowering Huntington levels in the brain at the same level as it's lowering in blood. And so if the mouse model is correct when it comes to humans, it tells you, but looking at lowering in blood may also be an indicator of how much lowering you're getting in the brain, because they're very closely linked in the animal model data. And that's really important. And why is it important? Because you need this data to be able to guide the correct dose. So PTC518 has been studied in animals, and it has been studied in healthy normal volunteers. And those of you who've been in the HD field as long as me and who have been to lots of EHDN talks and EHDN meetings will have seen Anu Bhattacharya from PTC over the last 10 years present this program, from the early screening data right to the animal model data. Anu spoke at EHDN in 2018. She spoke at EHDN in 2016. So she's been around and talked to many of you about this study in animals, and now it's moving into patients. So this was tested in healthy volunteers, and that's what you do with a new drug. You give it to people with no disease. And the subjects took a single dose compared to placebo, and it was looked at safety and tolerability, and then looking at whether the drug put the stop sign into Huntington. They also then went on and gave the subjects multiple doses to mimic more what it would be like in a clinical trial. And again, they looked at safety and tolerability and measured the stop sign being put into the Huntington message. They also measured lowering of the Huntington protein. Also, when you have an oral tablet, many tablets that you may take, there is a food effect. So most drugs for high cholesterol, blood pressure, thyroid, numerous drugs have a food effect. And so you need to test the food effect in the stomach on a drug. And that was done by measuring the how the drug moves around the body and the effects of food. And also the healthy volunteers gave CSF. They all had a lumbar puncture, and that allowed measurements of the Huntington in the brain. And it also allowed to compare drug levels in the CSF with blood. So now this is being tested in people with Huntington's disease. And this is the PIVOT HD's trial. So we want to look at safety and tolerability, i.e., is this drug safe? Is it tolerated? We want to see if it actually does what it should do to the Huntington message and the Huntington protein in patients. And we want to see its effect on blood, brain fluid or cerebral spinal fluid, and MRI markers of Huntington's disease. So this study is using the new HD integrated staging system and is focused on those in stage 2. And this is the first study to use the new HD integrated staging system, which I think is really important, and is focusing on those in HD-ISS stage 2. So
can I interrupt you there? Can you just very briefly tell us what's new with this scale compared to how we've done it before?
I will, of course, Astrid. So the new HD integrated staging system, it's, and many of you will have heard me talk about this at EHDN, is a way that accurately stages the Huntington's disease so it can be used for clinical trials. So what we haven't been able to do in clinical trials in Huntington's disease is have any clinical trials until people had really marked features of disease. But what we want to do is to go earlier in the course of the disease, because we want to try and see if we can possibly delay onset, slow progression, and have the biggest impact on the disease as a whole. So the HD integrated staging system, which is a huge amount of work, and as I say, is a subject of a talk in itself, which I would very gladly give you for those who couldn't go to EHDN, allows us to do earlier phase clinical trials. And it's going to really revolutionize clinical trials because it allows us to treat and potentially do clinical trials across all of the phases of Huntington's disease.
Thank you.
So
Really.
this study is going to be looking at stage 2 HD-ISS. And so you may not necessarily have what we used to call in clinic a diagnostic onset. And actually, the HD integrated staging system gets away from all those terms because none of them they're very useful for when you're seeing people in clinic, but for research and comparing clinical trials, it's very difficult to use terms that are very subjective. So what are the inclusion criteria? You need to be 25 years and older. You need to have genetically confirmed Huntington's disease. And importantly, the trial is, at this stage in the study, recruiting people with 40 to 50 CAG repeats, 42 to 50. You need to have a total functional capacity score of 13, a UHDRS independence scale of 100, and the PIN, which many of you may have heard of, is a mix of age, CAG, symbol digit, and total motor score. And what this study is having a PIN score criteria to have a very homogeneous cohort in stage 2. That means a cohort that are all fairly similar. And the PIN score is being done as part of the screening for this study. So importantly, these scores are helping the sponsor, PTC, to find HD gene positive individuals, but who have no functional decline. The total functional capacity and the independent score is completely normal. This is different from other recent Huntington's trials. And actually, this is why this is important, because it's using the HD integrated staging system, which was designed for this. Now, the PTC is using the PIN-HD to find the right participants. And the reason this is important is it tells us about the risk of a participant's Huntington's disease symptoms progressing, and higher scores mean a faster, more predictable progression. It allows you to have a cohort of subjects that you are able to understand more about how they would be expected to progress. So the HD integrated staging system is being used in this study. There is also plans to do a stage 3 study, which will be in patients with CAG greater than 40, biomarker of pathogenesis, and signs and symptoms with some functional change in the total functional capacity and independence scale. So it isn't just going to be in stage 2, but will be expanded to include those with stage 3. So this is the study design. It includes a randomized treatment period to either PTC or placebo. So the subjects undergo screening, and then they're randomized to PTC518 or placebo. And 2 patients receive PTC518 for every 1 receiving placebo. So it's 2 to 1. There's going to be a data readout at 12 weeks, which is going to guide the continued dosing. Now, stage 2, HD-ISS stage 2, and including now, which is new from the EHDN presentation that Amy Lee gave, including early stage 3 patients, they're going to be randomized to the same doses. The trial has started, and we has already dosed people in Europe, and a long-term trial is to follow. So the subjects we're in this study are comparing for 12 weeks data readout, then follow up continued for up to a further 12 months. So it's comparing part A, placebo or 5 milligram tablets, Part B, which is placebo and 10 milligram tablets. This 12-week data readout will tell us if we need to do Part C, which is placebo and a different dose, because what we have learned from many studies in Huntington's disease, it's all about delivery, but it's all about dose. So we need to be sure of the dose for any studies moving forward. So study visits, and this is an overview. Visit 1, subjects are assessed against the study entrance criteria, which is called eligibility at screening. And then once all that's been done, about 1 to 2 months later, if the participant meets eligibility, they undergo the baseline investigations, and they're randomized to whatever study is a part of the study. Part A, 5 milligrams, part B, 10 milligrams, or placebo, and part C will be a different dose depending on the 12-week data in part A and B. And they will begin the assigned treatment once daily. Visit 3 through to visit 5 is they return every month until visit 5 while continuing the treatment. And visit 6 and through to visit 8, the participants return every 3 months until visit 8. Now, visit 8 is the 12-month time point, and that's when the assigned treatment is stopped. We have a visit 9 and a visit 10, where participants are contacted for safety follow-up 1 month after stopping their assigned treatment, whether it was 5 milligrams, 10 milligrams, or placebo, and then again at 4 months. So this is the overview. Screening, baseline when you get randomized to placebo or active drug, safety assessments, MRI, lumbar puncture first dosing day, and then monthly follow-ups for safety and some of the clinical measures, and visit 8, which will be safety efficacy assessments, which are the clinical measures, MRI, CSF, and then safety follow-up at month 13 and month 16. So the screening assessments are really there to establish eligibility. They provide medical and medication history, physical examination, an ECG, an eye exam and photograph, blood draws, laboratory testing, and completion of clinical scale evaluations and questionnaires. The baseline period includes blood again, MRI, a lumbar puncture, and movement tests with a digital wearable device, which is a bit like a Fitbit. During the treatment period, people will have physical examinations, a heart scan, ECG, eye examinations, and a photograph of the back of the eyes, blood draws, laboratory tests, a lumbar puncture for collection of brain fluid, movement tests with a wearable like the Fitbit, and clinical scale evaluations and questionnaires. And also there will be an interview for potential side effects, which is part of the safety monitoring. And the assessments during the safety follow-up, including an eye examination and a photograph of the back of the eyes, blood tests, laboratory tests, and an interview about potential side effects, which is part of the safety monitoring. So I'd like you to thank you for your time in attending. I'm sorry for the technical problems at the beginning, and I'm very happy to take any questions. And the team from PTC who are running this study are also on the call, and they would be very happy to take questions as well.
Thank you so much, Sarah. That was really excellent and very well delivered. I could understand basically every word you said. I could understand. So thank you for that. Very, very helpful. People from PTC were here to be able to respond to questions or comments that may come up from you. So Brian Beers is here. You are global project leader, as I understand. Brian, can you please introduce yourself and tell us a little bit about what your responsibilities are and your role?
Sure, no problem. Yeah, I'm Brian Beers. I'm based in the US in Nashville. And as Austria mentioned, I'm the global product lead for PTC518 and Huntington's disease. So it's a pleasure to be with you here today. Yeah, we're very excited about this trial. We're very excited about the fact that this is now open at a number of sites in Europe, and really looking forward to increased participation and ultimately the study results. So happy to take your questions. Before that, I'll also introduce my colleague, Dr. Emily Bredlau, who many of you may have already seen here recently at the EHDN meeting. But Emily.
Thank you. I'm really happy to be here. Emily Bredlau. I'm the clinical lead for the HD programs at PTC. And I've been actually working in Huntington's disease since 2010 for most of that time. And I'm just thrilled to be at a company that's so invested in this community.
Thank you. So great to have you here. So really now the concept is that we will have a more open conversation about this and any questions or comments that you might have. And I think we already have some from the chat. I don't know, Tina, if you will help bring up some or.
Yes, in fact, I raised my hand because there are a couple of questions from Albert. Firstly, he wanted to know, and I think this comes up with all trials, would it be possible to unblind the study and stop placebo if the first results prove to be positive?
Yeah,
I think I'll give that to the sponsor.
Yeah, thank you. Agreed. It's a great question, Albert. And so yes and no. So no, we will not unblind the study. But what was not part of this presentation and is part of our plan is that we'll have an extension study to follow this. So anybody who goes on the study and completes the 12 months of therapy can go on to what is usually called an open label extension study. The study will not include placebo, but it will still be blinded. So if you were on 5 or 10 milligrams, you'd stay on the same dose and we would not unblind. But you'd stay on study drug. And if you were in placebo, you'd be randomized to one of the doses that are selected for that study.
Thank you, that's clear. And just one other question while I have the floor, also from Albert, why not try from a CAG of 36 rather than 42?
You posted that question when we were on the HD-ISS slide, I noticed. So I'm curious if the question is about HD-ISS staging or about the trial itself.
Albert, you can unmute yourself.
Oh, it's just a general question because 36 is just a threshold from where the transmission can happen and more largely. And people with 36 would say, like me, are susceptible to evolve towards the disease. And why not try as soon as possible and thus starting from 36 instead of 40?
I'm happy to answer that. So Albert, it's a very good question. And what's the HD integrated staging system recognizes the 36 to 39 group. But as you know, some people with 36 to 39 don't develop the disease. And what determines whether you develop the disease is still not clear. Because everyone with 40 or more, repeats, will always develop the disease, whatever happens, as long as they live long enough. That's what the HD integrated staging system. Now, people with 36 to 39 can have Huntington's disease. However, they have to have also a biomarker of pathogenesis, which could be volume loss in the brain that's very specific. It could be abnormalities in total motor score and symbol digit, or it could be functional decline. So they have to have 36 to 39 plus one of these measures because many people don't develop the disease. Lots of trials at the moment, even those that are not using the HD-ISS, are using 40 or more. Now, for the reason is that it allows a more homogeneous population, or a more population that looks more similar to each other. And the sponsor might be able to add something in here as well, if this moves forward and works out, then of course, it will be expanded to people with 36 to 39. But I think for the beginning trials of any drug, there has to be a focus. Now, the 42 to 50 is because this is a small study. It's a safety study primarily, and the other question is do they see any evidence of lowering the Huntington message protein? And this all has to happen first. And the reason the group is 42 to 50 is that we know how that group potentially will progress. And so you can compare it to historical control data. So it's a very long answer to a very good question.
Oh, thank you. But it's also part of a general strategy looking how to take care of rare disease patients.
Yes, yes.
Because, of course, we know Huntington starts in utero.
Yes, yes, exactly.
And so the most important is to start with a full life, a total life care, starting as soon as possible. And it's my question is also part of that argumentation that we carry towards the authorities, Minister of Health,
yeah,
etc.
no, you're absolutely right.
Thank you. Your answer was very clear. Thank you, Sarah.
But I guess what the inclusion criteria does is to maximize the chances of really seeing a potential effect if it's there, isn't it?
Exactly. And also over a 1-year period.
Exactly. Okay, Tina, you had your hand, but now you disappeared. No,
do you want
Tina.
me to
I
go
said,
through
I meant Dina. I meant Dina. Sorry, you had your hand up.
Sorry, I'm here.
Yeah.
Thank you, Sarah, for a great presentation. I was hoping you could clarify something because I'm actually a bit confused. So you stated in the inclusion criteria that TFC was 13 and UHDRS was 100, which is normal outcomes for the disease. But then you said that's the new staging system was stage 2. And in stage 2, is that not people who are already showing some sort of symptoms, signs and symptoms in the disease? So can you be in stage 2 and still have a TFC of 13 and a UHDRS of 100?
Yes. So the stage 2 group, the cutoffs for the stage 2 group from stage 1 are the cutoffs to be in stage 2 is a total motor score of 6. Which is low, but it is the cutoff that was from the normal controls gene, people who don't carry the HD gene with a CAG of less than 36, and a score below a cutoff from normals on the symbol digit task. So those subjects, other than something very subtle, are fine. They don't have any functional problems. Their independent score is 100%. Their total functional capacity is 13. So that's the HD-ISS stage 2. But what PTC have done is they have decided to expand the trial to include those with early stage 3, which means those with a change in the TFC and the independent scale that allows them to go into mild stage 3. Now, that inclusion criteria for that part of the study is going to be shared, but it is a change in the independent scale and the TFC. So it's going to be in stage 2 and stage 3, and PTC will be rolling that out. I don't know if you want to add anything to that, Amy Lee.
It was perfectly said.
Okay.
Thank you.
I think I'll, if I
Do you want me to go through the chat, Astrid?
Yeah, or I can just bring up Vlado's question. Is there any idea how much it will reduce mutant huntingtin and wild-type huntingtin? You did mention something about 50% or something, Sarah, from my studies, if I remember correctly, but.
So the mice studies show a very nice correlation with blood and brain, with lowering about the same. In terms of what's predicted with the 5 and 10 milligram dose I'll let PTC answer that. I think the question for me would be, does it lower it at 5 or 10 milligrams at all? And if it does, how much does it lower by? Which is the key question of the study. So, but I'll let PTC answer that in more detail.
Sure, I can jump in. So in the phase 1 study, after we dosed the healthy volunteers once a day for 14 days, at a dose level of 30 milligrams, we were able to reduce that messenger, so the messenger RNA, the messenger, by about 65%. So we know that we can go at least that high with, within that dose range. So then what that told us was that the doses, we need to select for this longer term phase 2 trial, they need to be below that. And so what we're doing, the approach that we took is starting low on dosing, because we've never dosed patients, we've never dosed humans for any longer than 21 days. So you start low and you make sure you know exactly where you can get to after a certain period of time. And we believe that certain period of time is about 12 weeks. So that's really the approach that we're taking. We're going 5 and 10 milligrams here initially to look and see where we get for the total Huntington lowering. And then we'll decide if we need to go higher than that to get more lowering. But as you know, we don't quite know in Huntington's disease what that magic number is. And that's really the substantive aspect of this dose ranging trial, is we're evaluating a range of doses to determine what works best in Huntington's disease.
Yeah, and I'll just add to that, because I saw Albert's note in the chat. Mouse models have shown that if you lower total Huntington by 30 to 50% in the brain, you can reverse some symptoms or slow progression. So that is the range that is aimed for, but because also it's lowering wild type Huntington as well. And I think from the data, there is no reason to think that mutant and wild type will be different with this approach. So let's say that they're going to be the same. Whatever is measured for mutant Huntington lowering will be the same for wild type. So that's exactly, Albert, why it has to be carefully dosed, because you want enough of the bad guy, but not too much, because you want to keep some of the wild type Huntington for important reasons. So that's why the different doses are so important. So the 30 milligrams was tested in healthy volunteers and gave 60% lowering, but the 5 and 10 in patients with Huntington's disease, we'll see how that works in terms of the dose. And also there's the option for the third dose. But that's exactly why this is a safety study and measuring how much lowering is got with each dose. I don't think there's any reason to think that the HD patients will be different from the healthy volunteers. But as Brian said, that's a 14-day study. So we have to just use that to inform this. And that's why the 12-week readout, which the sponsor will do, will inform them about the continued following 9 months, and also will inform about whether they want a third dose.
So can I ask you, as a doctor, Sarah, because maybe you are treating not only HD patients, maybe, are there any examples?
I don't treat anyone else, but
okay, but maybe you can answer still, or somebody
yeah, I will,
else
of course. No, I'm still a doctor.
Yeah, I know. So are there any examples of from the human brain demonstrating that we can really manage well with much lower protein levels than what would be kind of a normal? Because there are some concern about, we need the wild type. I think we all agree about that.
Absolutely.
But we don't know exactly how much we can manage with. Are there any other examples of proteins that people can manage where the levels are really
Well, it's
reduced?
A really good question for the whole field of everyone working with brain diseases, because many of these brain diseases that we work on are caused by levels of a toxic protein causing damage. And many of the approaches are working by lowering the levels of the toxic protein, but also lowering the levels of the non-toxic one. For example, in Alzheimer's, there's trials to lower the level of the amyloid or the tau, but it also lowers normal amyloid and tau. In motor neuron disease, the recent trial was lowering the normal levels of the SOD1 protein as well as the abnormal. So this is an important question for the field, and it's an important question. We can go by animal data suggesting that 50% lowering appears safe. We don't know, and so one of the things I think that's going to be important about this study is if the PTC518 turns out to not have any safety issues or minimal safety issues, then, and the dose can be really carefully managed, then this will tell us as it undergoes longer term follow-up, because there aren't issues of the way it's delivered. We just know it's a small molecule, but it will tell us a bit about wild type Huntington lowering safety, I think. So that's my personal view, in that we don't know yet. And also we have this fine balancing act where we have to lower the bad guy, which we definitely know is bad, but not too much because of the wild type Huntington. But I mean, this is a big topic about therapies for Huntington's. To target the mutant Huntington only protein is really, really difficult, because if you look at them both, they're essentially the same except for the CHE size. They're exactly the same. And so targeting something that just targets the mutant at the moment depends on these genetic flags, and they're only in a subset of the population, and which means you can't treat everyone, and they're really complicated. They may not be as effective. So we're in this fine balancing act, but many other therapies are in development for this, are in the same you know, if we had a magic wand, what would be the best drug, Astrid? It would be something that targets mutant only. But, you know, it's like we were saying, drug development's tough. And so we have to do it bit by bit.
And thank you, really. But I'm also thinking, I've heard that in Parkinson's disease, the level of dopamine is really, you know, being lowered throughout the disease progression, but it takes a long time. It can go really low before you have really clear symptoms.
Exactly.
So the brain works well, even if the levels go down quite considerably. That's
well, also we know from studies in Huntington's patients is you've got, and like Albert said, you've got the mutant gene from birth. And actually the brain works really, really well for a very long time, even with that. So we know the human brain is able to compensate. Yeah. And that's why we want to try and potentially treat earlier, because the compensation mechanisms may be just better.
Mm-hmm. Tina, please.
Yes, there was a question in the chat from Patricia that I didn't want to miss. She was asking about the rationale for the eye examinations. I spotted that as well. Why are you doing eye exams? Are they...
So one of the reasons for eye exams is that these drugs that are called splicing modulators of messages, basically, that put in a stop message, stop sign into the message. One of the, this is in animal models, is there is a question of if it can affect your retina. Now, this has not been seen in animal models with PTC518, but it has to be tested because it's a theoretical risk of the class of drug. And that's why the eye test and the photograph in the back of the eye is so important, because it's like every drug has a class risk. And a class risk means, so we know aspirin has a class risk because it can give you bleeding. So that's a class risk of that class of drug. We know that this is a potential class risk, so it has to be tested. And that's why the eye tests are being done so carefully, if that makes sense. And I don't know if Brian wants to add to that.
No, that's perfect, Dr. Tabrizi.
And there was
safety,
no problems with the
safety, yeah.
No problem.
Sorry.
No problem with the healthy volunteers, and they had their eye tested as well.
In the 2 weeks, Brian, do you want to answer that?
Yeah, so no, in the 2 weeks we didn't look for it in the healthy volunteers, but there was no need to test it in the healthy volunteers. It's really, they got 1 dose and then 14 doses. So this is the first opportunity, like we said before, where we have a longer term serial dosing where you expect to see, or could potentially see something from prolonged exposure to a molecule. So yeah.
Thank you.
Mhm.
Safety first.
Okay. Any other comments or thoughts about this? I don't know, maybe you said something about the timelines or whatever. You said you had started in Europe, Sarah, but can you elaborate a little bit more? Where are we in terms of
Well,
having people in?
I'll let the PTC answer that, because
Yeah.
we're recruiting. We've dosed patients already, and other sites are. We dosed the first patient in Europe. I'm very proud.
Congratulations.
I was waiting for that to come out.
But everyone is up, really moving on this. It's a really important study. So I'll let.
But maybe, yeah, please. Can you repeat which countries and how you're doing?
Brian and Amy, do you want to say which countries are involved and how people in the European community can get involved? Because I think the reason for this webinar is to let people know about it and get involved if they can.
Yeah, absolutely. This is a really exciting time for the trial. So it's been a long time. Clinical trials are not easy to get started. There's a lot of regulatory work that needs to be done. There's a lot of infrastructure that we need to put in place in order to get trials running efficiently across the world. And now is that time when everything is coming together here at sites across Europe. So just a quick rundown of the sites that we have that are activated in countries. So France, we have now sites activated, UK, obviously, Netherlands, Germany, and we also have a couple sites in Australia. And soon we'll be working on getting additional sites up and going in Spain, and looking at other countries beyond that as well. So, more to come on that. We don't have confirmation exactly of where those are going to be, but we are expanding quite a bit in Europe. So like you said, the best resource, or actually I didn't say this, but one of the best resources to look at to identify sites that may be closest to you is on clinicaltrials.gov. And also on the PTC website, we have a link basically to clinicaltrials.gov, which will populate our internal website, and you'll be able to see which locations are performing the trial.
Yeah, exactly. And I think the trial is important because the European part of the trial is super important, and we have an opportunity to, in Europe, to get ahead on this trial, do it properly, see if it's safe, see if it lowers the protein and so on. So I think, and because it's an oral tablet, it's just, we hope it will be easier for patients and families.
Did you say how many participants you are recruiting or aiming for in Europe?
Brian, do you want to, or Amy, do you want to answer that based on your stage 2 and stage 3, mild stage 3?
Yeah, so stage 2, which are the PIN score and the TFC of 13, IS of 100, 162 patients total are anticipated there. So a lot of those slots are still open. And then we're looking at approximately the same number of stage 3, but that addition to the protocol is still forthcoming. We have to complete writing the protocol and then get it through all of the oversight bodies before sites are able to enroll there. So there will be a total of more than 300 patients.
Mm-hmm. And it might be that by the time the stage 3 study gets up and running, the mild stage 3, there will be, I don't know timing wise, Brian and Amy Lee, but there might be 12-week data from that helps inform that. I don't know when the 12-week data will be. I mean, you have to wait till you've got 162 people, so it's not going to be super quick.
Excuse me.
Yeah, that's right. It's tough to forecast that at this point, but over the next couple of months, we'll have a really good indicator of how many participants are enrolling in the trial, and probably be able to better forecast that type of stuff.
And I think, Claudia,
and then
sorry, I just wanted to say, because there should be information about this, the trials on hdtrialfinder.net.
yes,
Isn't
that's
that correct?
right.
Yes, sorry. We had it. Dina and I have been working on this, and we actually have the page of for this trial, but it's getting updated today with this information. So thank
So
you.
please, if you could put just the web page address in the chat, that would be really nice.
Oh, sure.
Yeah.
My-
Sorry, I interrupted you, Sarah.
no,
Just wanted to have that
I
out.
just was saying, Dina, and then there's a couple of questions in the chat from Philippa, Julia, Julio, and George Yorling, which I think will be useful to answer. So Dina.
Actually, Astrid stated part of what I was going to say, that about the information on Trial Finder, because I wanted to correct Brian Beers from PTC, and asking any patient or carers to go to clinicalgov.com is the wrong thing to do. It's the wrong website for families. It is a very confusing website, and you may have the correct information on there, but it's not in lay terms for families to understand. So I just thought I'd feed that back to the sponsors about that, and that they should go to our trial finder, but Astrid's already stated that. So
That's why
a very
we
good
put it on,
point.
because it's easier to navigate for the lay community. So Sarah, you said there were a couple in the chat you wanted to pick up on.
Yeah, so Philippa Julio said that as we move to trials that are easier to participate in, and also earlier for people to participate, what impact do you expect on the HD community, especially the younger generations from HD families? A very good question. So this is exactly why the trial is moving into earlier HD-ISS stage 2, which is before the traditional what we know as onset, and but also will be including those with symptomatic manifest disease as with the new stage 3 cohort. I think that the importance of this, Philippa Julio, is to test if the oral tablet is safe, and does it lower the level of the Huntington protein, and in a safe way. If that all works out, then there will be, I'm sure, plans to do studies in stage 0 and stage 1, which is the younger generation from HD families. We aren't ready to do stage 0 and 1 trials at the moment because we don't have endpoints for those trials. However, if it all works out, and I think we have to remember this is a safety study and a dose finding study, so I don't want to get ahead of things, but if it did work out and it showed some benefit in HD-ISS stage 2, then if you could link that to a biomarker that linked to the clinical benefit, there would be the potential to go much earlier if the regulators will agree on that biomarker. So you don't need to do a 10-year trial. So that's a really complex answer to your question, but basically this is work in progress. And the younger generations from HD families is very much at the fore of everyone's minds in terms of this. And then the primary outcome of the stage 3 study be the same as the stage 2 study, safety. I think yes, George, unless PTC, it'll be safety and target engagement, i.e. lowering the level of the Huntington protein. And I think importantly, it'll be comparing safety in HD-ISS stage 2 versus mild stage 3 and see if it's different at all. Because all the studies to date have been in HD-ISS stage 3, and we may find that safety is better in HD-ISS stage 2 for all the reasons I was talking about compensation, but also will tell us if there are things that are common to both groups. So I hope that helps. I see Claudia put the hdtrialfinder.net in the chat. I think that's really important so everyone can see where it's being done.
Yeah. Okay. Thank you. That
Lots of
hour went
questions.
quickly. That hour went very quickly. And thank you so much, really, Sarah and Brian and Amy Lee, for being here and sharing all your knowledge and expertise.
Not at all. Not at all. I think
Really exciting times.
lovely to
isn't
see
it?
you all
Yeah.
and talk again.
Yes, absolutely. Absolutely. So thank you.
Thanks,
Take care,
Astrid, for
everybody.
organising it.
Thank you,
Thanks
everyone.
for the
Thank
amazing
you.
questions.
Thank you.